lenticels are usually they appear on the woody plants. It may be the dicotyledons or it may be the gymnosperms. Fine. I mean, in the plants which are in the lenticels are restricted or confined only to the woody plants, especially the gymnosperm woody plants as well as the angiosperms woody plants, usually the dicotyledons. Even sometimes we can come across, we can see even it on the monocotyledons such as on the bamboo. They also, but the occurrence of lenticels are very restricted to the woody plants of gymnosperms as well as the dicotyledons of the angiosperms, but very rarely, very in less number or less quantities of the monocotyledons may show the appearance of the lenticels. Right. So let's say, so this is on the what we call mature tree trunk, and this is what this again the white colored coloration what you are going to see. So this is the brown colored lenticels, and this the white colored lenticels what you are going to see it on the young. Now, I mean, again, this is also woody plant, but this is not yet matured enough. I mean, it's not a old one. Again, the green coloration, we can see that nothing but the young woody plants. There we can see the white colored lenticels. There, again, the coloration of the lenticels again is quite different. Again, usually the lenticels may appear in the brown or a golden brown color or in the dark brown color, but the formation of white color lenticels again is just because of the accumulation of a suberin. I will let you know in the coming slide. But when you take the section of the lenticels, I mean, if you take the transverse section of a wood, there you can going to see the opening like this. These are the lenticels, right? Usually the formation of a lenticels can be seen when the plant at the secondary growth state. Nothing but when when the tree or when the woody plants start forming the periderm, I mean the cork as well as phellogen, phelloderm, and phellum. There you are going to see the outermost part, that is what a bark, where you are going to see the openings or breakup of the outermost layer, and that particular region will be composed of the parenchymatous cells with a large intercellular spaces, and that's what we call it as the lenticels. That's what here we are going to see. Okay, then we'll explain in detail about that. Now, as I already told you that the lenticels are the again porous tissue, nothing but where you are going to see the openings that resulted due to the what we call the parenchymatous cells, which are having a large intercellular space on the periderm. That is here you can see that there is the parenchymatous cells which are having the large intercellular spaces also, and there is a pore. Here you can see opening, right? And these are the parenchymatous cells. You can even see the what we call the greater intercellular spaces. And as like usually what happens, the formation of a lenticels begins when the plant at the secondarily thickening region, or nothing but when the plant at the secondary growth region, where we can see the secondary thickening is forming at the time where we are going to see the upper outer covering layer, that is when the plant at the young stage or the primary growth stage, where we can see the stomata and the stem will remain open in the daytime and remain closed in the night time. But when the plant reaches the secondary growth or a secondary thickening state, there you are going to see what we call the epidermal region is going to be completely vanished off because of the continuous development occurred from the internal. Nothing but there we are going to see the secondary cambial activity going to happen, and vascular cambium, I mean, that is what the vascular cambium activity may happen, and the cork cambium activity may happen because of the pressure exerted, I mean because of the growth exerted from the internal cells. What happens, the epidermis is going to be broke down, and in the place of epidermis, there we are going to see the cutinization as well as the formation of the different cells. That's what we call it as a phellum, phellogen, and a phelloderm. Phellum is nothing but an outermost layer which has been formed by the cork cambium. That's what we call it. There is phellum. Next we are going to get the phellogen. The next is phelloderm. So because of the phellogen, nothing but this phellogen is considered as the internal, I mean the intermediary tissue between the phellum as well as the phelloderm, where this phellogen is also considered as cork cambium. Nothing but in outer division leads to the formation of phellum, and inward division leads to the formation of the phelloderm. So because of this, as well as in the there we are going to see the vascular cambium internally. If this is what a vascular cambium at the center, I mean towards the pith, where here we are going to see the formation of the xylem, and here we are going to see the formation of the phloem, the secondary phloem. So what happens? Phellum, phellogen, phelloderm, secondary phloem, vascular cambium, and secondary xylem. Like that we are going to see. Where what happens during such developments, where what happens epidermal, as the epidermal cell are going to be vanished or gets damaged, during that time the stomata are also associated with the epidermal cells, they are also going to be vanished. In the place of stomata, where we are going to see the formation of the lenticels, where we already know that the stomata remains open in the day or light, and in the night or in the dark condition, where the stomata will remain closed. With the in the most cases, where in case of lenticels, the lenticels will remain open for until they remain. Okay, there is no such closing and opening mechanism. We can see it in the lenticels as like that. We can see it in the stomach, where what is the function of this particular lenticel is that, now, as we already know that the stomata, which helps in the exchange of gases as well as the transpiration of water or vapor, similarly the lenticels also perform the same function, where they provide the pathway for the direct exchange of the gases, and those gases may be oxygen or carbon dioxide or water vapor. Okay, so when the plant at the primary growth stage, the stomata, what the function of the stomata, which will be continued by the lenticels when the plant at the secondary thickening organs. Nothing but when the plants start forming, when the wood, it may be a stem or it may be what we call root. Usually the lenticels are formed only on the stem region. What happens? These when the stem start secondary thickening, there the stomata will be vanished. I mean, the stomata will get broke down along with the epidermal, and in the place of the stomata, where we can see the functioning lenticels, they perform. I mean, they provide the chance for the wood, that particular bark or stem, to have an exchange of gases, and the gases function which is quite similar to that of the stomata. Where apart from this, the structure of a stomata also play a vital role, as like in the trichomes, how we can use the trichome structure and the shape for identification of a plant. Nothing but the trichomes in taxonomy. Similarly, the function or the shape of a lenticels can also be employed for the identification. Nothing but there are some plants or there are some tree species which have their characteristic lenticel structure, and that can be used for the identification of a tree plants. This particular slide will understand how the structure of a lenticels. I mean, now what are the components of the lenticels? How the lenticels is going to be formed? I think what the development of a lenticels. So usually, what happens? These lenticels, as we already know, that they are usually formed on the woody stem. Usually, let me tell you one more thing clearly, that even the root shows the secondary thickening, even the stem shows the secondary thickening, but the lenticels can be seen only in the arboreal or a terrestrial or a aerial portions of a plant. Nothing but underground portions of the plant, which though they have the secondary thickening, will not be having the lenticels, because there we cannot see any exchange of gases. Similarly, the stomata are not seen on the roots, right? There we can see the stomata on the leaf as well as the stem. Here are the lenticels can be seen only in the stem, where the leaves cannot have the lenticels, because the leaves cannot show, they doesn't show secondary thickening, or they doesn't show secondary growth formation. I mean, the bark formation or phellogen or the cork or such formation cannot be seen in the leaves. So, okay, and coming to the lenticel formation, usually what happens, as I already told you, that the lenticel formation usually begins from the beneath portion of a stomatal complex. Nothing but if the stomata is working during the primary growth condition of a plant body, if the stomata is working beneath that, the lenticel initiator will be there. Okay, so when the epidermis is going to be vanished, these lenticels will come into action to perform the gaseous section. During the time, consequently, this what happens, as there is a development of a lenticels, what happens, there is a periderm formation also be happening. Nothing but as the epidermis is going out of the plant body, along with the stomata is going out of the plant body, what happens, the stomata is going to form. I mean, pardon, excuse me, these lenticels are going to be formed. The lenticel formation begins from the first periderm formation. Nothing but the first bark or a cork formation, outermost secondary thickening covering. And the function, I mean, the what we call the occurrence of the lenticels may be in a different pattern. They may be circular, they may be oval, or there may be elongated areas, and they may be found on stems and the roots. Let me tell you again that if the roots are aerial, there only we can see the lenticels. Structure looks like it may be the rough or the cork-like structures and the young branches. And they what happens on the these porous tissue. I mean the openings or the what we call broke portion with nothing but a porous, which creates a large interstitial spaces between the cells, and through the space only we can see the exchange of the gases. And let again, as the first statement indicates you that the formation of the lenticels arises as a cell division forms as the cell division begins in the formation of the what we call phellum as well as phylloderm. Nothing but phyllogen is also considered as the cork cambium. This particular cork cambium initiates the formation of a phellum. So that's what the upper layer in the woody dicotyledons or the gymnosperms. There you are going to see the breakage of the epidermis. There the lenticel formation can be seen. Where the stomatal, where usually these lenticels can be seen usually not I mean only alle irutte anta ಹೇಳ್ತಿಲ್ಲ, usually they can be seen in the stomatal ground tissue region where the stomata were there you can see. Apart from that also the remaining areas also the lenticels can occupy their position. And now coming to the usually the color of the lenticels are again quite brown or golden brown or the dark brown in color, but sometimes we can even see the white coloration in the lenticels or completely discoloration of a lenticel that we have seen in the first slide. So that is because of the excessive deposition of the lignin in the cell walls of the lenticels. So that makes the discoloration or white coloration of the lenticels. Is that apart from the presence of the lenticels and the woody dicotyledon or gymnosperm plants, the specialized lenticels can be seen on some different parts of the plant body. So usually some of the examples here are given. So in the gray plants or in the whitish plants, where the presence of a lenticels can be seen it on the for example the base of a pedicel. I think you might have heard of a pedicels may have the lenticels, and that particular the function of a lenticels in the grape is to control the temperature. And they may also profound it on the some of the fruits. It may be the apple and the pears. Not all the apples may be having the lenticels, and there are some apples which will again the Australian or South American apples have these lenticels, and even the pears also. I think you have already seen the pears fruit, which can be sold in the different markets. In Kalburgi itself, there you can see such dark brown colored dots and the white colored dots on the apple. Can you all able to see these two images?
Yes, sir.
So actually what happens, these two plants, fruits, once after the formation of a fruit, they require high exchange of the oxygen. So because of that, the fruits have the intelligence of forming the lenticel, so that it can easily what we can have the exchange of the oxygen as well as the gases. So these two plants will show, especially the fruits will show the formation of the lenticels. And they may be also present on the potato tubers. Can you all able to see? Though it is underground, again let me remind you that this is though it is underground, because they require what we call when they are kept out of the taken out of the soil, they require oxygen to breathe. I mean the exchange of a gas should occur. Without the if they fail to exchange how the exchange of a gases, they become what we call a rotten, or they may become brown or black in color. So to avoid such what you call failure, the plants will have the intelligence of forming some specialized structures to have the exchange of the gas. And next one is the mangrove plants. In the mangrove plants, there you can see the nematophores or the breathing or respiratory roots and caritae. So in the mangrove plants, so usually they in the nematophores, there you can see the white color dots. Can you all able to see this one? Conventional woody plant, what they contains lenticels. Apart from that, some of the examples I would like to give you here. In the grapes, we can see it on the pedicel, and in the fruits, we can see it on the apple as well as a pear plant. And on the potato tubers also, we can see the presence of lenticels, and even on the nematophores, I mean the respiratory roots of a mangroves, they also contains these lenticels. Though it is not clearly elucidated the formation of the development of a lenticels, but I will by taking some of the images through the internet, I would like to give some information about the development of a lenticels. So can you all able to see this particular A image? This is A, B, C. It goes like this: A, B, C, D, E, F. Fine. This what can you all able to see this A? So this is what a plant which is after taking the section, transverse section. So a single plant. After the what we call after some days, as we can see, there is what we call there you can see the formation of what's the cork cambium, the initiation of a cork cambium results in that the cavity formation, as well as you can see the intercellular spaces. These are the parenchyma cells. Intercellular spaces are going to be seen. Once the pressure exerted, I mean, as the thickening, as we can see, the phylogens start dividing, the region which is going to be swollen, right? The erupted, we can see that. Finally, the eruption region, there you can see the enlarged cell. The cells are going to start enlarging. Nothing but the phellum is going to enlarge. Fine. So once they enlarge, we can see the eruption will be there, or the protuberance will be there. There again, we can see the cavity or a pore. Finally, we are going to see the crack forming, the initial crack, because of the continuous pressure exerted, because of the growth, internal growth. Then what happens? It is going to be broke down. So that particular crack, you can see it on the epidermal region, and this epidermal crack is again increasing their width. Finally, where it is going to become a cup-like structure. Fine. Can you all again one more point? You just observe that this again the green colored cuticle. I mean the epidermal is there. Again this epidermal is there. Again this here you can see the epidermal, but you can see the epidermal is losing. I mean the strength of epidermis is gradually coming down. Can you all able to see here also? Right. Again the cuticle deposition will begin. Right. The cuticle deposition will begin in somewhere else, and finally you are going to see the what you call the many layers which are going to form it on the cuticle. Here is the many layered cuticle. The secondary growth region, this complete region, and the finally we are going to see the cork cambium divides and complete the phellum is going to form. Finally, we are going to see the break of the complete cell wall, outer epidermal region, and finally you can see the lenticel formation. So this is how the lenticel or forms. Nothing but simultaneously what happens during the primary growth condition, the primary developmental stage of the plant body. There we cannot see the formation of a lenticels, but as and when there is a consecutive growth in the formation, whereas the vascular cambium will start forming the secondary xylem and the secondary phloem, and the cork cambium will start forming the phellum as well as the phylloderm. What happens at the central region of the word? What happens? The pressure exerted on the outer side of the outer epidermal region. That because of the pressure, epidermal will get collapsed. And finally, through the lenticels also going to be formed. So this is what the lenticels.